Good morning and welcome. For those of you who are coming just for today's seminar, welcome to a special seminar by the Center for Clinical Trials. My name's Kay Dickerson. I'm director of the center. We're extremely fortunate today, and I see you realize that too, because I see a lot of new faces in the audience from other departments and across the street, in having Dr. Janet Wittes here. She's a visiting scholar in the Center for Clinical Trials for the last 2 days and has been wonderful talking to our students and the faculty when we shouldn't, but we get words in asking questions ourselves. And it's just been a fantastic resource for us, and thank you for being here. So as part of her visit, she's agreed to do this seminar for all comers. And so let me tell you a little bit about her before she begins, and then she'll take up most of the time. She sent a bio to me, which is just so modest, but I'll go along with it because she's worldwide fame, and for good reason. She's a wonderful teacher and very knowledgeable about clinical trials. She's president of Statistics Collaborative, her own company that she founded in 1990. One of the main activities of Statistics Collaborative is that they serve as a statistical reporting group for independent data monitoring committees for clinical trials, mostly multi-center trials, I believe. Previously, she was chief of the biostatistics research branch at National Heart, Lung, and Blood Institute, and that was from 1983 to 89. And then she wrote her 2006 monograph, Statistical Monitoring of Clinical Trials, a Unified Approach, with others, and that deals mainly with sequential trials. She's also written quite a bit, and I'm sure in this audience you've read numerous things that she's written. Her research is focused on the design of randomized trials, the capture-recapture methods in epidemiology, and sample size recalculation, which may be part of what she talks about today. She served on a variety of advisory committees and data monitoring committees for government and industry and several FDA advisory panels. She was formerly the editor-in-chief of Controlled Clinical Trials, now called Clinical Trials, and served in that role from 1994 to 1998. She got her bachelor's degree in mathematics from Radcliffe College and her PhD in statistics from Harvard. I give you Dr. Janet Wittes.
Well, it's really nice to be here and to see some faces that I haven't seen in a long time, and then those of you who I've never seen before, and some that I met yesterday. There are a lot of you with whom I've worked over in one capacity or another, and so it's very nice to see you all. So this is a little bit of a false advertisement. These cautionary notes on adaptive designs. False because the subject is so broad, and as I was speaking to Milo last night to find out, who you were going to be, and to try to make sure that I addressed the issues that would be relevant to the audience that was likely to come. So we'll see whether that's correct or not. But I'm making the assumption that I should go back here before I start. That most of the trials that you do here are not adaptive in the sense that the aficionados of adaptive talk about. The push for the kind of adaptation that I will talk about at the end of the talk comes mostly from industry, I believe, where there's a real desire to do trials that get finished quickly and to develop drugs rapidly. And so that's the push for adaptive designs. The trials that you do in schools of public health are really quite different. They're looking to change treatment, like we just learned about PSA and selenium. I guess it was vitamin E today. Big changes in public health trials. And so the kinds of adaptive designs that have been very that have been in the statistical literature recently are really not mainly what you do, but I will talk about them anyhow. So the pitch that those who are advocating adaptive designs, there's lots of seats here if you don't want to sit on the floor back there, that clinical trials are too rigid, that there are new classes of designs and they're called adaptive, and they save money, time, and so forth, and therefore they're more ethical because they're faster, because you get treatments to patients faster. But we need to be, and the lingo is adaptive by design. And what I will try to do is say I disagree with most of this. I don't think clinical trials are too rigid. I don't think that most of the designs called adaptive are actually new. I think they risk. If they work, they will save money, time, and so forth, but they may not. And I will say that in many cases, you don't have to be adaptive by design. But I'll try to say what in some ways I agree, in some ways I disagree. But that's really the pitch. So I want to start with an example where failure to adapt led to failure. This was a study, and I'm still not at liberty to say what the study was because of under confidentiality, even though this was years ago. The confidentiality is probably gone, but I don't know. I was on the DSMB. It was a high-risk cardiovascular population on standard of care, so that was the background therapy. About 10,000 patients, so a big trial, randomized 1-to-1 to the new treatment or placebo, and the outcome was basically MACE, CV, cardiovascular death, MI, or stroke. We anticipated that, so both the designers of the trials, although they didn't really tell us that, but the DSMB anticipated that, that there'd be very little difference between the placebo and the treated for about a year, and then there would be diversions. So we expected that we were going to have to sit tight with our fingers crossed as we saw nothing for a while. But we said, and then this is for the statisticians among you, we don't really care that from an analytic point of view, we don't really care that we don't really believe in proportional hazards. We're going to use a log rank test anyhow, because although it's not optimal, it has high power even in a case like this. So that's what we were thinking. The data monitoring committee, we were a very experienced crew, and we knew each other very well. We had sat together on other committees, other cardiovascular trials with the same group that was this was a McMaster group that was putting it together, the Population Health Research Institute, the Salim Yusuf's group. And we knew how the other ones thought. So that was very comfortable that we didn't have to learn about each other through the course of what turned out to be complicated and difficult decisions. And we also believed as a crew, although my friend Leroy Walter says don't use the word belief except when you're talking about religion, but it comes out anyhow. What? Faith-based, right. Faith-based conservative bounds. But we really all felt that we could live with very conservative bounds for efficacy because we didn't want to have a trial stop early and then, somebody says, oh, well, you didn't wait long enough to find the adverse events, or, this may have been true in Romania, but it's not true in the United States and stuff like that. So we were very comfortable early on saying that the bounds were going to be really conservative for stopping. And the other thing that we as a group felt, and not everybody feels this way, but all of us did, we didn't want any safety bounds. We said the Potter-Stewart thing, we'll know it when we see it. If it's if it's bad, we'll pull the plug. If it's not bad, we won't pull the plug, and don't tell us what to do. We'll just figure it out. That sounds more hostile than it was. It was actually, everybody was in agreement with that. So, and we expected to see harm before benefit, because we expected there was going to be bleeding and other bad things, that the harm was going to be, this was a treatment that was going to be given continuously over time, and we expected the harm risk to be pretty constant, and that there would be no benefit seen, as I say, for the first year, and then there'd be benefit. So we expected to be sitting there with bad things happening and no benefit seen, and we were going to sit. So that was the context of the trial. Now, and there was a brick wall between us and the sponsor. So the sponsor saw the pooled event rate, and they saw that all along. Now, some people don't like that. I mean, if Tom Fleming will say the sponsor shouldn't be even seeing that, but they did, and we had no objection to that. And we saw the event rate by group throughout the trial. So there was conversations, that we lobbed across the wall, but we always lobbed, when we talked to them, we were always thinking about, well, no, we weren't. We were always pretending that we were thinking about the pooled event rate, and they were always trying to elicit from us what we were seeing separately. Okay, so what did they see? They saw the rate was half of what was expected. Well, we saw, and so that got them nervous, because they thought, oh my goodness, there's not enough events, and therefore we've got to increase the sample size because the event rate is lower than we expected. What we saw, yeah, that was right. Overall, the event rate was half of what was expected, but the effect size was about 30% greater than expected. So that was pretty exciting, but the power was low. All right. So what happened? They asked us.
Sorry, when you say power?
Oh, the condition of power.
So the effect size you were actually seeing?
The effect size we were actually seeing. Yes, that's a good question, Dan. Yeah, anybody, please interrupt me for clarifications. Yeah, we were, what we looked at was the conditional power given the overall event rate, or the two event rates, and the observed 1.3. So even with that, the power was low. So they asked us, should lobbing over this wall, should we increase the sample size? And we said, we won't tell you. Now, you all understand why we won't tell you. Because if we told them, that would give them, then they would know something about, they could figure out, all they knew now was the event rate, the pooled event rate was low. But if we told them to increase, that would make them think about various scenarios that might be going on. So they interpreted the "we won't tell you" as, aha, things are looking good and we shouldn't increase the sample size. And we said, darn it, to ourselves, because we really wanted them to. We really felt that they really, this drug was probably very good. And if they didn't increase the sample size, we're going to see nothing. Oh, what did I do? Okay.
Even the event rate being half, they wouldn't think to increase the sample size?
They figured that our failure to answer was saying that the effect size was so huge. So the calculation they did was that they figured that their standard of care arm was what they figured. And then there was this magical effect size in the treated group. That was the calculation they made. I mean, now you can make all kinds of faces you want, but think of, you're the sponsor, an industrial sponsor. The way PHRI works is that they do the study, they run the study and they have autonomy, but there is a sponsor behind them, and an increase of the sponsor, which is a company, that increase in sample size would have to be paid for by the company. So the company was really making that decision. And I'm going to use the word belief again, or faith-based. When companies do trials, and when you guys do trials, even though we use these words about being what's the word we use? Equipoise, yes, that we're in equipoise. You really believe this stuff is going to work. I mean, that's because otherwise, why go and do 10,000 people and millions of dollars if you think, yeah, it's just a coin toss. So you really believe it. So you see something like this, you see a very low event rate, and the DSMB, that you think, this is experienced committee, says, don't do anything. You think, aha, I've got a magic treatment.
You think don't do anything. You said we won't tell you. We're not gonna...
We said we won't tell you. They interpreted it as, no, that's a really good point. We thought we were very clear. We won't tell you. We didn't say, we won't tell you guys because don't you remember we're a DSMB and we're not supposed to tell you. We didn't say that. We said, we're not going to tell you. We thought naively that was sufficient, that they should have understood that that's the purpose of the wall, that we're not going to tell you things. But you're right. It was a gift different in what we said and what they interpreted. So after the trial was over, and it just ended when it was supposed to, the effect size was about 10% higher than assumed. The event rate was so low that the p-value was 0.13, and the trial was called a failure. Now, I never liked the word failure for it, but in a sense, this was a failed trial because it probably was an effective drug that wasn't shown to be effective because there were so few events. So at the end of the trial, when the data came out, the head statistician of the company called me and said, why didn't you tell us to add sample size? And I explained to him as best as I could the dilemma that we as a committee faced and how we felt that we couldn't tell them, but that we had hoped that they would understand that our not telling them was not informative. So what was the lesson? Clearly, adaptation of the trial would have been good in this case. We had no prior guidance, and part of that was, I guess, our own hubris. We said, we don't need any prior guidance. We're experienced. We can do whatever. We'll know what to do. Everything was done post hoc. Had there been some discussion in the protocol about under what conditions the trial there could have been an increase in sample size, maybe there would have been. I mean, who knows? It's very hard to know counterfactual. And if the sponsor had increased the size on the basis of the pooled event rates, there would have been, some people would have wagged their fingers. And I'll give you an example of finger wagging. If you shouldn't have done that. You should have done exactly what you said you were going to do. But I think, my opinion is, but we don't know, of course, that the trials likely have shown benefit. The moral of the story to me is that if you're going to change the protocol ad hoc, which is what would have happened, everybody must really understand who can see what. And the failure, I think, here was that the two sides didn't really understand what they were. Well, we understood, but they didn't understand what it was that we were allowed to say and allowed to do. So this is an example that weighs heavily on me because I think it's likely that a good treatment bit the dust. Yeah.
When you were doing these interim analyses, you have to. So you weren't doing information-based monitoring.
This wasn't this was not information based. This was fixed time. And
If you were doing information-based monitoring, you would have run the trial longer.
Right.
And you would have.
Yes. This was done in an era, 10, 15 years ago, where the typical trial was not done information-based. Did you all understand the difference? Okay. You can run a trial in one of two ways. It's probably one of 50 ways, but the two ways that we're talking about is, one, you say, I'm going to run the trial for this amount of time. And you fix the time, and you estimate up front how many events that are going to occur in that time, and then you figure out the power that way. Information-based monitoring says, or time says, I'm going to run the trial until I have 320 events. And you follow until that number of events, and then as Dan says, you would, in that case, the trial would have naturally extended longer. Many of these trials now that are done, especially in cardiovascular disease, but also in cancer, a lot of trials where their endpoints are clinical outcome events, binary events, are done as information-based trials, and this would not have happened in that case.
It might still be going on now.
Yeah. I'd still be behind the wall. So here's two other stories. The first one is a trial that I'm involved in. These are very short stories. A trial that I'm involved in for a very small company, a really interesting prevention trial. And I've been working on the protocol with them, and I get the name of the protocol, and it says an adaptive phase 3 trial comparing blah, 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 blah. And I call the medical head, and I said, what do you mean adaptive? This isn't adaptive. It's a regular trial. It's got two arms, and we're randomizing. He says, if I don't use the word adaptive, the venture capitalists won't support this trial. So I said, okay, I promise you during the course of this trial, there'll be some changes. We'll call that adaptive, and I'll make you an honest man. So it sits there as the part of the title. The other one really bothers me, and this is a trial that I'm not involved in. I was involved in, but I pulled out. But it's fairly typical of the results of the advertisements about adaptive designs. This is a trial in cancer. Think of a really bad cancer. It's that. If you can think of a really bad cancer that has really high mortality, that would be this one. The drug that's being used has been studied in like 7 people in a phase 1 trial. Nothing. And the assumption is a 50% reduction in mortality. And I said, you can't assume 50% reduction. I know you think this is great, and you've got these mice and these rats and stuff like that, but, 50% reduction just doesn't happen. And the answer was, but we have a method. If we assume 50%, we have enough money for a trial with 50% reduction. But what we'll do, we know it's not going to be 50%. We're going to use one of these methods, one of these adaptive methods sold so highly in the, and we're going to look at the trial in the interim, see what the effect size is, and then we're going to go to the venture capitalists and with the real number that we need and get some money. I said, okay, fine. You go to somebody else. I'm not playing this game. But this is one of the dangers. This is one of the dangers that I see, that when you use a method for a reason that it's not designed. This is a kind of a reason to get money and to fake people out and just imagine how the IRBs are being, how the optimism is being misrepresented to the IRBs, which, interestingly enough, didn't object to this. I've been amazed. Didn't object to, my goodness, how could you be assuming a 50% reduction in mortality? But I've heard this a lot in many very small companies. So what's wrong with the pitch? Yeah. Wow, that's interesting. See, I thought it was restricted to the little tiny companies who were, wow, that's interesting.
Janet, what is like Cytel selling in terms of adaptation? What are they trying to sell? Are they trying to sell, look at the effect size?
Yeah. Look at the effect size. I mean, there are different things in the package, but the main, well, we'll get to that, okay? But yeah, the main pitch is look at the effect size. Okay, so what's wrong with the pitch? One, I'm going to contend is not really so new. It doesn't always save time or money, and the designs may be much less efficient than, statistically less efficient than if you designed it correctly. The answer to that is, yeah, but we don't know what the numbers are, so how can we design it correctly? So that's an issue in the literature that's been going back and forth. And then there's a lot of discussion about being adaptive by design, that you can only, if you're going to adapt a study, you need to say up front what kind of adaptations you're going to make. You design them in. And I'm going to say you don't always need to design up front. But in some cases, it's essential. So I'm going to be, on the one hand this and the other hand that. And to me, there's four kinds of adaptations. There are operational ones, which I'm going to argue we all do all the time. There's those that are fixing nuisance parameters, and those that are changing the hypotheses. One, on the basis of external data and second thoughts, and two, on the basis of internal data. And we'll go through each kind because I think they really are inferentially and theoretically different. So to me, an operational design, you sort of titrate the design. You add and drop centers, you encourage adherence to study protocol, you change the entry criteria, things like that. So you have a design, you have a hypothesis, but you're titrating it to get it to be more efficient and less noisy. And then there are changes that you make in response to external data. So the two that I was most familiar with there were in the studies of antioxidants after the carrot study and the Finnish smokers study, where the antioxidants, the smokers increased mortality. Smokers on antioxidants had increased mortality. Studies that were looking at antioxidants at the time kind of pulled smokers out. So that was an adaptation to the design in response to external data. That seemed to me, just how could you not do that when you had replicated scary data? The Women's Health Initiative, the initial design of the study had defined who was going to get opposed and who was going to get unopposed estrogens. After the PEPI study came out and there was bleeding, they changed the criterion, and so people who in the past would have gotten opposed estrogens got the other way around. So those are changes. You get external data, you learn more about your population, you learn more about the risk, you change the study. That seems to me perfectly fine. And, how do you write that by design? I mean, you write the only sentence you can write is, if something surprising that affects us happens, we're going to change it. I mean, that's a sort of a silly sentence. So to me, this is not so new. It's like Monsieur Jourdain and Molière's Bourgeois Gentilhomme. Good heavens, for more than 40 years, I've been speaking prose without knowing it. I mean, we all make changes, and we do it, and we don't think of it as particularly surprising. Then there's the fixing the nuisance parameters. And this is where, what are the nuisance parameters? There are the variance, there's the underlying placebo rate, or the control rate, which would be in the first example that I gave you, the non-adherence rates are kind of nuisance parameters, things like that. So you could do a prior study to find those events, but what we all know is that the events that you see, the event rate or the variances that you see in a single center study, a small single center short study, they're just plain different from what you see in a large multi-center, multinational long study. So those estimates that we get they're good to start with, but we don't really believe that they're going to, or maybe I should say it differently, we need to be cautious about how convinced we are that what we're going in estimating is what we're going to see.
So Janet, one comment, I think those are gained also, and maybe even with the appearance of more safety than when you gain the endpoint, because you can have an optimistic sigma squared and say, well, we'll adapt, get the venture capital, instead of what I would say you should do is to be somewhat pessimistic and then adapt if you need to be able to make it shorter. But the gaming there can be just as extreme as for the hazard ratio or something like that.
Oh, I totally agree. Absolutely. Yeah, and of course, I mean, I think many of us are guilty of that. I have to confess to, sometimes you've got a sample size and, there's a budget, and the only way you can get the sample size to work is to make the variance a little smaller than you think it really is. And you sort of, clutch your guts and hope. And then you say, well, we're going to do some longitudinal analysis. That'll change the variance. We'll take an extra measurement. That'll change. I mean, so there are things that, even those of us who pretend we don't game things, you do, or one does, or I do. So what, I'll skip that because, so if you're paranoid, as I tend to be, about what you can learn from other data, you're worried that pilot populations are small, they're relatively homogeneous, the estimates aren't reliable, the variances are too small, the pilot hazard rates and hazards aren't accurate. And then even in safety, you only see, if you have a small study, you're only going to see the frequent events. So you're going to be underestimating, you're very likely to be underestimating the harms. And the only hope, I mean, this is the real pessimist in me, in looking at efficacy besides the rats and the mice, is looking at surrogates and markers, because your sample sizes are going to be just too small to get much of a sense of efficacy.
Now, when you interact with pharmaceutical companies, they come with you and say, we need to do sample size, we need to do a study of size 500, you help us justify that? Or, it's a different funding, NIH grants, they're of X amount and you've got to fit it in the budget. Is it the same thing with pharmaceutical companies, or they give you a little bit more?
It's the same thing. Yeah, because they have a budget for the whole drug development scheme. And so, yeah, I mean, there's a certain amount of back and forth as there is with NIH grants, but you can't, if you come with a size that is out of the ballpark, there's no way that they can fund it, or that they will fund it. So anyhow, you make up these data, right? I mean, that's what we all do. Nathan Mantell used to say, I design trials as with bricks without straw, which is, of course, from Exodus. And here's one of my favorite. Whoa, hold the phone. You're telling me this whole time you've been just making up the horoscopes? So with internal pilots, you basically start the study with unanswered design questions, which we always do. You don't know the variance, you don't know the event size, you don't know the effect size, and you look at early data and change the design parameters. I will skip this because, all right, sorry. So when you don't know the variance, you can look at the data either a blinded or an unblinded way. There's methods for both, and it's looking halfway through is probably not, in general, a bad time to look. And there are methods available to preserve type 1 error rate. So if all you're doing is, or more generally, if you're changing nuisance parameters, not the effect size, if you're changing nuisance parameters, there are lots of methods to do that, and they preserve type. You can do methods that preserve the type 1 error rate. Yeah.
Janet, I think it's important to point out that nowadays, I think people are getting away from relying on variances from previous trials more for covariance and then using the new trial in an enriched population based on what they've learned, those most likely to respond, get away with a smaller sample size with that. And or else, even if it isn't enriched, if we know the covariance, to do an adjusted analysis at the end where they might be able to get away with a smaller sample size.
Yeah. I take a pessimistic view about that because I'm always I think that's, if you can really do it, I think it's good. But again, very often those are based on relatively small sample size, looking at predictors in small sample sizes and assuming they're going to be good, which sometimes they are and sometimes they aren't.
Usually, though, phase 3 trials of a similar treatment that another company did. That's what I was referring to.
Oh, yeah. Yes, absolutely. So I would say the methods here aren't really controversial. In grants, they're hard to include that just because of the way granting mechanisms work. But from a methodological point of view, I don't think this is controversial. So again, the same sorts of things for proportions or survival. But now let's get to the more difficult issues. Changing the hypothesis. And I'm going to say it in that term rather than changing the effect size, because I think if you say, oh, we're going to just change the effect size, it sounds not sure we'll change the number. But if you say it as we're changing the hypothesis, it sounds more serious. So, and I want to distinguish between the basis of external data and rethinking, and then the basis of internal data. So here's an example from the SHARP study. And I don't usually like to have a lot of text, but I want you to see the actual words because I think they're important. So here's a study that it was Merck and Oxford had the control, where they changed the primary outcome. And these are the literal words from the website, the sharpinfo.org website. SHARP originally set out to assess the effects of lowering cholesterol on the risk of, quote, major vascular events, defined as major atherosclerotic events, plus other cardiac causes of death and strokes due to bleeding. But whilst, so you can tell it's British, the trial was ongoing, new information became available from other completed studies. And that's the important thing. It's external data that suggested that lowering cholesterol might have little effect on these additional events. Before breaking the trial's code, again, a very important thing, so they didn't know, the people who made these changes didn't know what was going on. Therefore, the independent steering committee decided that analyses of major atherosclerotic events would provide the most valuable information to patients and doctors. This change was described in a paper that was published in a leading medical journal before the trial's code was broken. And then the conclusion, taking the combination of zemitide and simvastatin long-term reduced the risk of heart attacks, strokes, and operations to open blocked arteries. This is for the public, that's why it's written like this, by about 1/4 in people with chronic kidney disease, irrespective of the severity of their disease. So SHARP, this is Colin Bayesian, was the PI. They made a major change in the endpoint, in the outcome, And that was the outcome that they used. Interestingly, Merck didn't accept that. So there's Sharp has two results. It has the Oxford result and the Merck result. And this is the description of that. If you're going to change the primary outcome, do it with trepidation. And this is again from the web. There is a certain incertitude, again, it's so British, right, concerning the primary endpoint, apparently changed by the steering committee before the trial ended. But this change was not endorsed by the sponsor. So what you have is a very public record of a change, not endorsed by the sponsor, and two interpretations of the data because the results were different. What the Oxford crowd did showed benefit, and if you added all that noise, didn't show statistical evidence of benefit. My own feeling is I agree with the Oxford people. It seems to me when you have external data that says, we're looking at cholesterol-lowering drugs and we're not seeing an effect and all this other stuff, that seems to me a good enough reason, and not only a good enough reason, a good reason to change the primary outcome. But I think this little quotation, and you'll see similar things when you see other discussions of this paper, tell you that if you're going to do that, and this is going to be true whether it's a public health kind of trial or a drug development kind of trial, you have to anticipate that not everybody's going to like it, and that there's going to be a lot of discussion and disagreement. So other similar changes that to me make real good sense, and I always endorse them, but again, cause problems with people who say, whoa, you wrote in the protocol this, and now you're doing that. You change the method of analysis, and especially in light of Dan's report, right? Everybody's changing their method of handling missing data. Well, not everybody. People who used to do the wrong things are changing their method. So switching from change scores to longitudinal analysis, which I think a lot of us are doing, as you recognize why. You have all these observations in the middle, and you're ignoring them, and you're not reducing the variance. That doesn't make any sense. So there's lots of changes that you can make that weren't anticipated in your original protocol or your analysis plan, that you, as you think about what the analysis is, makes sense to make and are independent of the data. You're thinking of them through just what you know about analysis.
You can accept that change to longitudinal analysis may be mostly motivated by missing data that make it difficult to compute change scores. So, yes, that's right. Not totally independent, but right, but independent of the effect of the, again, done blinded to. So not independent of the data, but independent of knowledge of the treatment code. Okay? But now they're changing the hypothesis on the basis of internal data. So, and here I have, as you can probably figure out, I have two minds. I often have two minds about things, and it's very hard for me to come to I'm a on the one hand, on the other hand kind of person. For those of you who believe, there's that word again, that the way you're supposed to choose a hypothesized effect size is to say, what is the clinically relevant difference? If that's how you do things, then that's a hypothesized delta, okay? Now, I don't do that. I do sort of the other way around, that says, what do I think this intervention is going to do? That's my hypothesized difference. Do I care about it? Oh yeah, then I'm going to do the study or not. But so it's philosophically somewhat different. It probably comes out to be the same, but I don't, it's very hard for me to know what it means to say that this delta is the clinically relevant difference. I just never know what that means. So,
Weird.
what?
It's weird.
It's weird? What's weird?
I mean, so we're doing a study, right? I mean, I'll often involved with where they have some kind of continuous measure, right? So some psychometric measure, and they don't know what a 1-point change is. So if you don't know what a 1-point change is, should that be your primary endpoint? I mean, if you don't understand the endpoint and its clinical relevance.
But if that's what, let's say you did understand is clinical relevance, how do you translate that to what is the clinical relevance in the population at large? I mean, that's the blood pressure question, right? What matters to the individual is really different from what matters to the population at large. So I just don't know how to think about it.
We're doing population, I mean, our inferences are about the population, not about the individual. Even though we try to then prescribe drugs based upon the results of this study,
right?
Which is drawing population level inferences. We're not saying anything about the individual.
Right, but when you say what is the clinically relevant difference, I don't know what. I have a hard time. Do I care if my blood pressure reduces by 2 millimeters? Probably not. Do I care if the population does? Probably yes. So anyhow, this is a different topic, but let's say that the way you design something is you understand clinically relevant difference. Okay, we have to start that way. So if you start that way and you're a frequentist, you can look partway through and you can define when, and it should be infrequent. That infrequent is different from frequentist. It's different. You calculate the observed delta and you recalculate the sample size. And that's basically what Cyrus's program is doing. That's what AdPlan is doing. It's looking at the hypothesized effect and changing the sample size. And there are many ways of doing that technically. The Bayesian way is analogous. You look partway through. You can do it more often because you're Bayesian. You calculate predictive power and you decide whether to stop or add more. So the problem that I have is if we chose the delta rationally, which I don't know if we did, but if we did, if you believe the delta is rational, then why is it okay to suddenly say, oh, I'm interested in a smaller delta because that's all I'm seeing? Because that seems to me philosophically to be saying, I didn't choose it because it was the clinically relevant difference. So to me, that's a change of a hypothesis. The other problem with the frequentist approach is that the methods, and maybe I'm exaggerating, I'm saying very inefficient, cross out the very, they're inefficient. Why are they inefficient? Well, they're inefficient in the following sense. If we know what an optimal design, an optimal statistical method is, that's optimal. So you have an optimal method. So you have a log rank test, say, for proportional hazards. So any other method, if that's optimal, must be not optimal and therefore inefficient. More specifically, what's happening in most of these methods is that you have some data, then you adjust, and then you collect more data. And the subsequent data are downweighted. So an observation that occurs after the adjustment counts less than an observation that occurs early. If the number that you have to increase after that adjustment is very large, then you have a very large proportion that are downweighted. And so that's the two-bit explanation of the inefficiency. Problem with the Bayesian approach is in ordinary sequential analysis, if you don't cross a boundary, you can't declare success, right? A boundary for efficacy. If you do cross a boundary, you don't have to stop. And if you don't stop, you recoup all the alpha that you would have spent. In Bayesian analysis, at least, and I'm talking about Bayesian analysis in the regulatory setting, must you follow the rules? I think true Bayesians say no, but I know the FDA says yes. And I've been involved in a study where the DSMB, of which I was not a part, thank goodness, refused to follow the rules because they said, if you needed us to follow the rules, why did you need us? And the FDA said, well, then the whole analysis doesn't count because you have to follow the rules because this Bayesian analysis said this is what you had to do. I don't know what's right or not right, but I've seen this and that has scared me. I don't.
Just being a true Bayesian, can I make a quick comment?
Yeah, absolutely.
True Bayesians would say you do have to follow the rules, even if it's just for face validity. But actually, it's by not following the rules, everything is, even as for a Bayesian, more fragile. I'll just leave it at that.
Well, let me tell you a conversation that I had, because I thought you had to follow the rules too, all right? So I was at a meeting, this was an FDA something or other workshop meeting, and there was a discussion of using Bayesian methods for adaptation. And I raised my hand and I said, well, the problem with that is you have to follow the rules and DSMBs won't follow rules. And the panel was up there, and Telba Irony, she's at the Devices. She's terrific, and she's really thoughtful, and she's a true Bayesian. She said, "What are you talking about? You don't have to do that." And then everybody on the panel agreed. Yeah, you don't have to do that. Go back to your, basically say, go back to your books, Janet. You don't have to. So I scratched my head and I thought, okay. And Don, of course, Don Berry had said, you have to follow the rules. So not being a true Bayesian, I don't know what the answer is, but I know there's a public disagreement about whether it's okay or not to follow Bayesian rules when you have a DSMB. So that's all. I put it on the table. I don't have any more to say about that. I don't know, and you guys will know this better than I. I don't know. My sense has been that the eagerness to adopt the adaptation methods is really coming from industry and FDA. FDA being more cautious than industry. FDA saying, fine, do this as much as you can with phase 1 and phase 2, but we're really nervous about phase 3. My sense is the NIH, or that the study sections are more conservative. I don't know. So the dilemma. I think external pilot studies or pre-studies are important and necessary. They're kind of boring. If they're really just pilots, they're hard to fund very often. They're internal pilot studies when you're using the data to re-estimate your parameters, are effective in tweaking parameters, but probably difficult for grants. The methods that are now considered more interesting, the ones that are popularly called adaptive, I think they fundamentally change the hypothesis if you really think that you chose the delta for rational reasons. They're statistically inefficient. They may not be so inefficient that you don't want to do them, but you've got to recognize their inefficiency and simulate enough to know how inefficient they are in the situation where you're in. And importantly, they require extremely careful protection of the blind, because if you have a study and everybody knows you're going to increase the sample size if the effect size is too small, and suddenly the sample size is increased, well, people aren't so stupid. They back calculate. So there's all kinds of ways that people do this by saying, well, we're not going to tell you the sample size. We're just going to tell you when to stop recruiting. So then people don't know that there's been an increase. Or you say, well, there's been an increase, but we're not going to tell you how much. So there's all kinds of ways that people do this in order to prevent back calculation of what must be going on. So I think failure to adapt when necessary risks failure. For operational changes, I don't believe you need a plan. For recalculating nuisance parameter, I think it's good probably to have your protocol have an intent to do this so people don't think, oh my goodness, they just made this up. But if you forget to do it, I don't think it's a disaster. Response to something that happens outside, you can't have a plan. But adapting on the basis of an observed effect, it adds power at the expense of sometimes a large increase in sample size and changing the hypothesis. Thank you. Yeah. Any questions? questions? Yeah.
I want to thank you for this excellent lecture, and I think it's very important to point out that there are adaptive designs that can be quite inefficient. I mean, in Wittes and Meta in 2002, they
Yes.
actually theoretically showed that there are certain types of adaptations, changing the sample size, that can always dominate. You can always do better with group sequential design. I just want to add a quick one story that when you talk about the NIH, how receptive they are to adaptive or not. I just have one anecdote. I'm not sure how much this can generalize, but I'm involved in a study with NINDS, and they really pushed us to consider adaptive designs. And there's a consortium. It's related, like very consultant groups connected to this. But there was a push from NIH on us to consider adaptive designs.
That's really interesting.
And I think, and the motivation was, can you make this more efficient? And I don't think it was necessarily because of the buzzword of adaptive designs as much, I mean, I'm hoping at least, as much as they just wanted to be, can you make this a little more efficient with some adaptations? And if not, it's okay. But if you can, that would be great. We'd like to save money in this current political climate. And I also wanted to point out that you had mentioned the efficiency or the inefficiency of some certain adaptive designs. And that's certainly true. There are some that are terrible. But there are also cases where adaptive designs can be more efficient than fixed designs. You can't do it if you assume you know everything, you know all uses parameters, all your assumptions are exactly correct, then you can't really be an optimal fixed design. But in general, you may not. There may be some uncertainty. That's what will happen, and then you can show in some cases that the adaptive designs do better than fixed designs. But not the adaptations you talked about, but changing subpopulations.
Yeah, no, I totally agree, and that's why I do think you have to simulate, because there are examples where you do much better, and so it's just very hard to generalize, and the concern that I have is selling designs for the sake of designs rather than saying, okay, here's our problem, what's the best way to design this study? Yeah, Jay.
I'll just add about changing effect size on the basis of an interim analysis. Usually a smaller than what was thought to be clinically relevant at the beginning of the trial. What the big pharmaceutical companies will do when they go to the advisory committee with that result is they'll have these high-paid thought leader consultants from industry come who will say, oh, of course the smaller effect size is important. If you'd asked me at the beginning of the trial, I'd have told you that. Meanwhile, they could go to 30 potential consultants. The 3 that agreed to say this are the ones that they bring. I call that lobbying, thought leader lobbying. Bad as K Street lobbying.
Tom.
Just another point on the adaptation or dangers thereof, and I'm an advocate of at least considering it, is the delta you've had on the screen at all times never had a subscript on it. But if we put a subscript T on it, very much like your first example where there might not be much going on early on and then there's separation, you've got to be very careful that even if the current delta hat is accurate, it may not be the delta that operates subsequently, and you don't want to lose power for the separation that's going to happen later.
Yeah, no, that's a really good point. And actually, the other side of that is it's not really relevant to adaptation, but it's relevant to pulling plugs on a trial too early when there's nothing for a while, and you just have to sit there and say, the reason we did this trial is we really think this intervention works, and we think there may be a delay, and we're just going to sit. And you never know. Once the plug is pulled, the trial is out.
Didn't you see that in the study that you were talking about at the beginning?
Yes, but we didn't pull the plug.
It was 30% higher than expected. At the end of the day, it was only 10%.
Yes. So that was the other way around. We attributed that. I mean, who knows? We attributed that to noise, but who knows? Yeah, Gary.
So there are other types of designs that find themselves under the name adaptive, like adaptive randomization, changing eligibility, which you did mention, to enrich for particular subpopulation. They don't necessarily correspond to sample size re-estimation. My sense is that there's a lot of interest in adaptive randomization as a way to perhaps either through ethical arguments or financial arguments to try to make the studies more efficient in a different way. But you didn't talk about the randomization stuff. So do you see that as something completely different?
Yes, I see it as something completely different. A whole different topic. I mean, the question is how do you the adaptive randomization is done, well, it depends. It's done for two different reasons. One is adaptive randomization is done to ensure balance across lots of different minimization. And that's what I think is different. The other is adaptive randomization. I think this is the one you mean, where you sort of pick the winner, where you change the allocation ratio on the basis of the effect size thus far. So no, that is not completely different, and I didn't think of it as I was putting this together. There are other kinds of adaptations that I didn't address at all. There are other kinds of enrichment designs. There are withdrawal designs. I mean, there are all kinds of and actually, what I was thinking of in terms of the entry criteria was something more simple than what you're thinking of. I was just thinking about you go in and you discover that these people that you thought couldn't be entered, you can, or these people that you thought was safe to be in the trial, they aren't. You're talking about a more sophisticated change, which is, this group of people seems to be doing better. Let's enrich the population. Yeah, I mean, there's all kinds of adaptations. But I think what you have to do is, as you're thinking about moving down from operational to changing hypotheses on the basis of what you're seeing, as you're moving down that gradient, the farther down you get, the more you have to be really careful that you understand the consequence of what you're doing, both to the trial, to the efficiency of the trial, and to its credibility. Yeah, Dan?
How should the point estimates and confidence intervals be?
Oh, I don't know. And there's let me repeat the question, because I growled at the question because I don't know the answer. The question is, in these kinds of trials, what do you do for the estimate? And boy, there's just a huge amount of disagreement about what you do. So I don't know the answer. How's that? Okay.
Well, thank you very much, Janet. Thank all of you. And although